Welcome to Carriganore House. This is the location of the European-funded CREST project, funding from the European Research Council. I'm now just going to show you around. Our Vision Centre, the Vision Research Centre, was opened on the 23rd of March by An Taoiseach, our Prime Minister, Enda Kenny. And this was a great occasion for the Waterford Institute of Technology and for the South East of Ireland to have this type of acknowledgement that the Taoiseach came to Waterford to open our centre. This is, as I've said, the location of where we're going to run the clinical trials that are being conducted as part of the European-funded CREST project. So patients that are enrolled into this trial, these are two types of patients, patients with early age-related macular degeneration, and we're also doing studies in the young normal population. They'll be greeted by one of our researchers and they'll be taken into the reception area, which, as you can see, is quite nice location for them to get ready and prepare themselves for the trial. At this part of the study visit, the patient will speak with the researcher, make sure that, again, they're fully aware of the trial, and we'll discuss the informed consent part, which is very important in a clinical trial, that the patients are fully aware of the interventions but also the tests that they're about to undergo. So this is the first part of the patient journey, the patient visit. This house also has access to a very nice boardroom, which is very important for group meetings, but also for meetings and discussions with the funding organisation, the European Research Council, and collaborators that are working and supporting with us on this trial. We have our weekly and our group meetings here, and also we can have patients in here where we can sit down, discuss the studies and answer any questions that they may have on the studies.
My name's Sarah O'Regan and I'm the Research Assistant on the CREST project, which is funded by the European Research Council. My role is to act as first point of contact for the patients and to organise study appointments. My responsibilities include ensuring that data is collected in an appropriate and efficient manner.
So this is the Vision 1 lab. This is where we measure vision in the clinical trials, and as I've said, at a super sensitive level. This is one of our PhD students, Sarah Pickett, and Sarah is going to tell you some of the ways that we measure vision, from standard vision charts but all the way up to measuring contrast sensitivity at different levels.
So the object of CREST is to take normal vision and make it better and enhance it to a higher level. And one of the first things that we will do on subjects is assess their refraction, their spectacle prescription, so that their vision is optimized before they go ahead and do any of the vision tests that we have. So starting from basics, really, the first test that we do on patients is their visual acuity, which is basically their ability to see letters black letters against a white background, which is shown here in this chart. This is something most people would be familiar with from going to their optician and getting checked for glasses. So we do that as standard on every subject for both eyes, with and without their glasses. We then use the same chart to actually measure contrast sensitivity. So contrast sensitivity is a little bit different. It's the ability of the eye to detect the foreground from the background. And so we're able to choose letters of differing contrast and then actually assess their vision at those levels of contrast. And so if you're able to see something very, very faint against a white background, your visual performance is actually better. So that's one of the ways in which we measure contrast. This machine here is called the Visual Function Analyzer, and it's another measure that allows us to assess a subject's contrast sensitivity. But in addition, uniquely, it's allowing us to assess glare and the influence of glare on subjects' vision. Importantly, all the measures that you'll see in CREST are actually non-invasive, which is very important for any trial that's assessing a large number of patients.
One of the interesting things about the CREST project is that the measures that we're performing are around vision, visual performance, visual experience, and at a sensitive level. But what we know in terms of the populations that we're studying, we have the young normal group who think their vision is optimal, but we believe we can enhance that. But when we look at the AMD population, what's interesting is that the measures that we're doing when people have AMD, these visual parameters and their visual function is actually worse. So there's consistency in terms of the methodology that we're doing in both trials. It makes sense that we do that. But we believe that whether we can halt the disease, whether we can prevent the disease, we're not so sure yet. But we do believe that we can enhance the visual function and visual experience in both populations under investigation. And one other test that we do is we look at photostress recovery, so how well one responds to a bright light source, such as a light source what we see here, the blue light source. And remember, macular pigment is uniquely located and has the ideal optical properties to filter out that blue light. So we've set up standardized tests that allow us to look at glare with the contrast device, but also photostress recovery, and we've set up standard methodology to allow us assess that. One of the important proposed functions of macular pigment is its ability to filter light, blue light, particularly 460 nanometers. Now we know that light scatter is a problem for vision, and because light scatter causes glare, and particularly blue light is what contributes to this. So this device here, the Sequant, will allow us measure in a standardized way light scatter and the effect of light scatter on vision and how that changes over time following supplementation. So we believe this to be a very important measure in the CREST project. So obviously one of the main objectives of CREST and what we believe we can achieve is that we can enrich macular pigment uniquely with all 3 macular carotenoids. And it's very important that we are positioned and able to measure that. And this is one of the devices that we use to measure that. It comes from Brown University. Professor Billy Wootton was the developer of this technology. And this uses a principle called heterochromatic flicker photometry. But it's been developed over the years that allows us to customize it for each patient, which is very good because the accuracy and reliability in the measurement is very good indeed. So at baseline we'll measure the patients, we'll get their spatial profile, and following supplementation at their preceding study visits we'll be able to measure this also. We customize the flicker and the eccentricity, the location at which we measure with this control panel here. But we've had great success with this technology over the last number of years. Okay, one of the important measures in the AMD trial that we're running in CREST is morphology. So what that means is disease. Do patients have disease? How are we going to assess the disease? How does that change following supplementation? We're working with the University of Wisconsin, Professor Ron Klein's group, to see about retinal grading. And there's now an internationally accepted grading system, the AREDS grading system, which we're going to use in this trial. So our big challenge in the CREST project is to get patients with early condition, and in such patients you'll see that their macula will have some drusen and maybe pigmentary changes. So we're going to work with ophthalmologists and the optometry community to try and identify patients that are suitable for enrolment into this trial. By doing that then, we will send stereo fundus photographs to the University of Wisconsin, where Professor Klein's group will grade them and confirm that they are indeed in that early age-related macular degeneration group. So that's really what's of interest here. And it's amazing that a camera like this can actually get us right back into the back of the eye, where we can really look and quantify the amount of disease that's present. Other technology that we'll be using in this trial is we're going to be using OCT. This technology hasn't arrived just yet, but we're using the Heidelberg Spectralis OCT, which will allow us to look at foveal architecture as well in a very unique way. And this is quite novel to the CREST project because it's possible that enrichment of the macular pigment may have benefits in terms of foveal structure and the cells required for vision. And we're going to try and look at that in a very sophisticated way in the CREST project. One of the other projects that is currently underway at the Macular Pigment Research Group is the EXIT study, and obviously we have a keen interest in understanding the role of nutrition for vision, and particularly how nutrition can affect macular pigment levels. An egg is a very good source of the macular carotenoids, but it's also a very good transporter and deliverer to the retina of the macular carotenoids. So we have a PhD study being conducted by Katie Marr underway that's looking at optimization of eggs and how these optimized eggs may enhance macular pigment levels and have an impact on visual performance. In addition, as part of this egg analysis, we're also going to assess other foods for the macular carotenoid content, and we have again a particular interest here in mesozeaxanthin because foods haven't really been tested properly to see if they contain mesozeaxanthin in addition to lutein and zeaxanthin, and that's because the assay and the methodology required to do so is very complex, very difficult, very expensive to run. There's probably only 4 labs in the world that can actually do it, and ours is one of those. This work is kindly supported by the Howard Foundation and Nutricite Consultancy Limited. So recent publications from our group have been the result of studies conducted under the MOST brand, and that is mesozeaxanthin ocular supplementation trials. These trials have been kindly supported by the Howard Foundation of Cambridge, UK, and again they're studies around mesozeaxanthin along with the other macular carotenoids and how these enhance visual performance, visual experience, and it's in fact this data that has informed the structure and the design of the CREST project. So without the data that we've collected over the last number of years, we wouldn't be at a situation that we can conduct the CREST trial in terms of the sensitivity of measurement, but also in reliability around of the number of subjects, the number of patients that we need to test for the coming years to prove this research idea. So this is Vision Lab 2, and again, in here we are able to measure vision in many different ways. It'll be a flow over room for us, but in addition, this is where a lot of the pre-screening will happen in terms of eligibility for inclusion into the CREST trial. Our ophthalmologist that will be working on this trial will screen patients who have been identified with early AMD or who have been informed that they have early AMD to actually confirm that. So we're going to put a lot of effort in the CREST project to make sure that the patients that we're testing fit the definition of early age-related macular degeneration. We'll use slit lamp assessment in order to do that, and then we'll combine that with the assessment from Professor Ronald Klein's group in terms of the grade and the stage of AMD that they're at.